Welcome to Nikolai's genetics lessons, and today we are going to talk about what is a single nucleotide polymorphisms, what is a haplotype, and how to apply SNPs profiles in order to choose personal drugs, what we call personal medicine. Look around at any crowd of people, and it's obvious that no two of us are exactly alike. Beyond our distinct physical appearances like height, hair texture or skin tone, there are hidden differences beneath the surface. Our unique biological makeup dictates our individual susceptibility to certain diseases and even determines how effectively our bodies respond or metabolize different medications. But where do all these variations actually come from? To find the answer, we have to look at our genetic code. DNA and genes act as the master blueprint, holding the precise instructions required to build and maintain a human being. When we compare different people, the root of our individuality becomes clear. It is written directly in the varying chemical sequences of our DNA strands. These strands of the DNA represent different chromosomes, just small portions, of course, of them, and these triangles shows different genes on those chromosomes. And in between, there is intergenic space that doesn't code for anything, but sometimes we can find regulatory sequences there that regulate how genes are expressed. By mapping human genome, scientists have discovered that our genetic differences aren't usually massive overhauls. Instead, they are often incredibly tiny, single nucleotide substitutions, essentially a single mismatched letter change in a vast sea of genetic code. In genomics, these specific point variations are called single nucleotide polymorphisms, or SNPs. Let's zoom into molecular level to see exactly how SNP functions. To illustrate this, let's isolate two short segments of DNA. Both of these samples are taken from the exact same chromosomal region, but they come from two completely different individuals. At first glance, the color-coded chemical basis looks remarkably similar, but closer inspection reveals a subtle twist. Take a look. CG here, CG here, TA here, TA here, AT here, AT here, AT, but here, in this individual, we see substitution of adenine with guanine, and this also leads to substitution of thymine with cytosine. If we consider only one strand of the DNA, one we call coding strand of the DNA, another template strand of the DNA, so single substitution we can find in one of these individuals. But the rest, as you see, bases are the same. So let me circle it. So here we have single nucleotide polymorphism. But does it mean that person on top doesn't have polymorphism? Actually, he does also have single nucleotide polymorphism, which can happen in different individuals with different frequency. For example, the top one can be found in 28% of the people, and the bottom one in 72% of the people. If we isolate just one strand of the double helix to keep our comparison simple, we can see that the sequence of bases is an exact match, letter for letter, except at the one solitary position right in the middle, where the top strand has adenine, highlighted here in red, the bottom strand features guanine, highlighted in green. This single letter typo is a classic signature of SNP. These variations aren't just random, isolated mutations, they propagate through the entire populations. For example, if we analyze this identical stretch of DNA across a broader population pool of 1,000 people, we would find a distinct demographic split. As this pie chart shows, 72% of the population carries guanine variant at this position, while the remaining 28% carries adenine variant. In bioinformatics software and genomic databases, scientists don't always look at the separate strands. Instead, they often stack the alternative alleles right on top of each other at the exact locus or location, creating a consolidated look at population's genetic diversity at a single glance. While single variation might seem small, the cumulative effect is massive. Genomic mapping estimates that the human genome contains at least 10 million of these unique SNPs scattered across our DNA, creating the incredible tapestry of human diversity. To see how these variations impact real-world traits, let's move to the next chapter. What is haplotype? While finding a solitary SNP is a milestone in genetic mapping, a single point variation isolated by itself usually won't reveal very much about a person's overall traits, health, or physiological responses. Because our biology is incredibly complex, looking at just one letter change gives us highly limited view. To get a meaningful data, we have to look at the bigger picture. To establish a clear, predictable association between genetics and something complex, like how a patient responds to a specific medication, scientists can't rely on just one location. As you can see, along this timeline of code, researchers must track multiple SNPs scattered across a much longer stretch of DNA to understand how these independent variations interact with each other. When you factor in multiple variable positions across a single gene segment, the math multiplies quickly. If you have 3 different spots on a strand that can switch letters, you suddenly generate a wide array of possible SNP combinations. These varying sequences cascade down, showing us just how many unique genetic arrangements can theoretically exist in single region. Take a look. For example, here at this locus, there are 2 letters that can be found. So we have 2 variants here. So let's put it here, 2. And here at this locus, we also see A and G. So we also have 2 variants here. And in the third locus, we also have G and T. So another 2 variations. So 2 times 2 times 2 give us 8 different unique combinations. For example, T, A, G, or it can be T, G, T, or it can be, for example, G, A, G, or C, A, T, and so on. In genomics, each specific combination of clustered SNPs inherited together on a single chromosome is called haplotype. In the isolated region shown here on the screen, the variations yield exactly 8 possible haplotypes, neatly stacked to showcase the distinct patterns of highlighted genetic letters. But here is where real-world biology gets fascinating. When scientists actually sequence DNA samples from a large population pool, they discover that nature doesn't use every mathematical option. In this typical example, the shaded rows reveal that 4 of the theoretical combinations don't actually exist in population. Instead, the pie chart shows that the vast majority of people are concentrated into just 4 active living haplotypes, with haplotype 1 claiming the lion's share. To understand how these are expressed in individual, we have to look at the heredity. Remember that human genetics operates in pairs. We inherit one chromosome containing a specific haplotype from our mother, and matching chromosome which we call homologous chromosome, with a second haplotype from our father. Combined, this child inherits a distinct dual-layered maternal and paternal set known as haplotype pair. Take a look at this human karyotype. As you see, we have 22 pairs of autosomes and 1 pair of sex chromosomes. So all chromosomes goes in pairs. One we got from mother side, another in each pair we get from the father side. So we inherit two haplotypes, maternal and paternal. Depending on what your parents pass down to you, your haplotype pair can take two forms. It can be different, like in this two loci, or can be completely the same as we see on this picture. If both parents pass down the exact same sequence, your haplotype pair will be completely identical. Ultimately, you can think of individual's unique haplotype pair as their personalized biological SNP profile. When modern pharmaceutical researchers or doctors analyze how a person will react to a specific drug, they look past individual mutations and instead evaluate this holistic SNP profile to predict efficacy and side effects. Now you should understand SNP profiles, and we can go to applying SNP profiles to drug choices, and we can talk about personalized medicine. To understand the practical application of this research, let's look at the common clinical scenario. Albuterol is a drug widely prescribed to patients to help relieve the acute symptoms of asthma. However, medicine is rarely one-size-fits-all. While albuterol provides effective relief for some, it fails to help others entirely. Scientists are currently hard at work studying why individuals with different SNP profiles demonstrate such varied responses to this treatment. At a cellular level, we know that albuterol functions by acting on specific receptor called the beta-2 adrenergic receptor, or beta-2AR, located on the cell membrane in the lungs. So here is the albuterol, and this is receptor. The beta-2R protein is encoded by ADR-RB2 gene, which you see here. To understand how genetic variation affects this receptor, researchers analyzed 3,000 base pair stretch of DNA encompassing the ADRB2 gene. Within this specific region, they identified certain distinct locations where SNPs naturally occur. By studying these variations across diverse populations, scientists successfully identified 12 different haplotypes, each representing a unique inherited combination of those 13 specific SNPs. As we've established, we inherit our genetic code in pairs, one haplotype from our mother and one from our father. Each unique combination of these two inherited haplotypes forms an individual personal SNP profile. And on this slide, you see a pie chart which illustrates the relative frequencies of common SNP profiles found in patients diagnosed with asthma, labeled A through E. When scientists tested the drug's efficacy against these 5 common profiles, distinct patterns emerged. Albuterol provides excellent relief for individuals with profiles B and E, while patients with profile C see only fair results, and most critically, patients with profile D show no response to the medication at all. So as you see, this research paves the way for more personalized future in medicine. Soon, a physician will be able to determine the patient-specific SNP profile and compare it against the data to accurately predict whether they will respond to albuterol potentially saving patients from ineffective treatments. It also comes back to foundation, the ADRB2 gene, which encodes the structure of the beta-2-RD protein. Small changes in that gene translate to the changes in the protein, which ultimately dictate how a patient interacts with life-saving medication like albuterol. I hope my lecture today gave you better picture and idea what is a SNP, what is a haplotype, and about personal medicine. And this is all for today. Subscribe and see you in the next video. Goodbye.